All right, guys, can you hear me? Let me double check that nobody's having trouble getting over. Whoops, in Zoom or Blackboard. It's been a long day, guys. I have been in classes since 10, like back to back, so I'm starting to lose my voice. All right, so we are going to do the lab. Oh my goodness, I need to record this. Hold on. Record. Oh, it is recording. Okay, good. I'm on top of it then. All right. Okay, so we moved over to Zoom because my Blackboard camera doesn't work, and I'm going to be showing you all my things today. So I needed to be in Zoom. I don't care if you're on the camera or not. I do want you to find Dahlia Johnson, which is my daughter's name, but also a picture of me. Okay, you should see double of me because this one is going to be showing you stuff in the microscope, and then I'm going to be showing you like big stuff on this camera. So you need to be able to see both of my cameras. Okay, well, here I'll set it up. All right. So for the lab, you can do the lab with me right now, or you can just watch and do it. There you go, move later. Okay. I'm recording this and I'm going to send it out after class today, so you can rewatch it, do whatever you need to do. If you are going to do your lab, you need your microscope. You need some glass slides. You'll need the microscope cover glass. And you will need the slide combo onion root prepared slide. All right, when you open it, it looks like that. And then you'll need your shrimpies if they hatched. I think I still don't have any hatchies yet. Sorry. Give me one moment to put my dogs outside. Thank you, Aiden. I always forget. Okay. If you're going to do the lab with me right now, you're going to need your microscope. If you're going to do it later, then you can just watch and you don't need it. So let me share with you the assignment pages that you need. The student guide looks like this. You can find it all over the place. You can find it in the introduction to 1.05. You can find it on the lab report submission page. You can find it in the email I sent you and the announcement. Okay, so should be able to find it. This is the directions. This tells you all the things that you need to be doing during the lab. And then the other one that you will need is the graded assignment lab report. Okay, so this one is where you answer the questions, and this is where you turn it in, or this is what you turn in. So we're going to be talking about these questions here. 1 and 2 we will answer in class as I talk, and then hi Jasper.
I just saw Wally at the store. Is it okay? I'll get a Wally.
You want to get a Wally? Okay. That's my toddler, guys. He just got home. I'm going to do a class though, okay?
And we can play it.
Okay. All right. Number 3, sketch some of the observations from your lab. You're going to be doing that. What you see in your microscope, you're going to draw it, or you can take pictures with your phone and send those to me. And then number 4 and 5, the answers you're going to find in your online lesson, the 1.05 lesson. Okay, there's pictures in there and answers if you read through it. Any questions about what you need to turn in or what you need to do right now? Awesome. I'm going to send this recording out too so you can double check it later. Okay? So go ahead and pin this video, not this one. I'm going to put my microscope one down. So the one you can see me in. So to start, here is your microscope, right? So you always want to carry it by the arm and the base because it will fall off sometimes. This is the eyepiece. This comes separately. This is a lens. This one is 15 times that you put in here. This is the eyepiece. This is the rotating lens thingy-mabobber. So the shortest one here is 4 times. It is the lowest power, but you have to remember there's also a 15 times here. So it's 15 and 4. Then the second one, the second longest one here is a 10 times, okay? And then the longest one is the 40 times, okay? So it's 40 plus 15. All right, down here is your light. This is where you get the light to come through. This little doobobby, it makes more light come through. So you can change it to have less light come through or more light. I always start with the most light, the biggest hole. Okay. And then I also always start on the lowest magnification. I haven't sent the email yet. The directions for the lab are on your lesson for 1.05 for the lab, but I will also be sending out some additional directions and this recording after class. So you won't have the email yet. Okay? Any other questions? Questions about your microscope or anything? If your shrimpies have hatched, they can go like a day or so. I don't think I have any yet. They can go a day or so, and then I would feed them like the tiniest pinch of flour. Like you don't want to put too much flour in or that it will kill them. So you can just keep them in the same area or the same jar, container, or whatever, and then just add a tiny bit of flour every few days or so. Awesome. I hope you guys have some because I don't, but I got some pond water with some living things in it. So there's some stuff swimming in around in here that I will show you on the microscope too later, since I didn't have any sea monkeys to show you. All right, so for the lab, to get started, you do not have to move your sea monkeys to a different container if you don't want to. Okay, whoopsie. There we go. All right, so your light is on, your microscope is on the lowest power. You have this guy in here up here. These little things are the slide holders, but they annoy me, so I don't use them. You can use them if you want. So first step in your lab is to use this. It's the prepared slide. So it's an onion root and an onion root nodule. So make sure you hold it on the edges. You don't want your fingerprints on it. So when they say an onion root, it's like, so if my finger was the root and they took a slice of it this way, that's the onion root. The nodule is if they took a slice of the root like this way, right? So across ways. So you have both of them on here and you want to take a peek at both of them. They are blue and pink because they've been stained, right? They're not normally that color. So what you're going to do is you're going to put it on your stage here. Try to see if it's in the light, okay? And then you're going to take a peek inside and slowly turn your focusing knob down. All right, you don't want to start way down, you don't want to start way up. I'd say just as long as there's a good gap in there. The problem with starting all the way down or all the way up is on your high power lens, it's a possibility that if you got too close, you could break the slide. So you're going to move it around, try to find that onion root. There it is. And then make sure it gets in focus. If it's hard to see, you can also adjust right here to have less light coming through. Sometimes that helps with the focusing. Okay. All right, so now I'm going to switch to this camera so that I can show you inside of my microscope. So I ordered a camera that goes inside your microscope, but of course it's not here yet. So we'll just have to bear with me while I find, there it is. Oh, wrong way. There it is. There we go. All right, so that's the slice of onion root that you should see. Let me see if I can focus a little better here. Okay, so those circles, the pink circles in the middle, those are not cells. Those are actually the holes in the middle of the root that carry the water up to the plant. Hopefully I can get back to it. Sorry. Be prepared for me to be doing this a lot in class. The last block we found a single-celled organism, so that was fun. All right, there it is. So that's the slice. Then you can also move it around and find the nodule. So let me do that right here on this one because they're hard to see. There it is. All right, so this one is pink. Hopefully I'll do better this time. Oop, there it is. Oh. Anastasia, I just saw in the chat that you don't have something. Is it the prepared slide that you don't have? So there's the root tip. Pretty cool. All the little dots are cells, the pink cells. Okay, so now to get a better picture, what you're going to do is you're going to switch to the next higher power. So you're going to move up to 10. Okay, again, make sure when you're moving it, it doesn't touch the slide. It should still be there. Of course, mine's not. There it is. And then you just adjust the focus a little bit, and you get a much higher magnification with that. Ugh, sorry guys. Next time we use microscopes, it'll be much better. I'll have my fancy dancy camera. Oh, I thought. There we go. Nope, maybe. There it is. All right, so you guys can do this too with your microscope and your cell phone if you wanted to take pictures of what you're seeing, or videos if you see sea monkeys that are swimming around. So you don't need the onion root tip to complete the lab. It's just one of the things you can look at. Okay, so don't worry, Anastasia, if you don't have this. So that's the 10 times power plus the 15 of the lens. Now, this is the tricky part. If you're going to move to the highest power, this one is so long that if you move and it's too close, it could break your lens or your slide. All right. So what I like to do is I like to just lift it up and then turn it because I have broken a slide before. Not a lens, fortunately, because those are expensive, but then you're gonna just slowly, slowly, slowly go back down. And I'm gonna tell you right now, the high power is really hard to focus. Even I struggle with it, and I've been looking at this stuff forever. So you just want to barely, slowly move around to see if you can find something. Ooh, I think that's it. Nope, that's the edge. What's this? Oh, well, I found some gunk on the slides, but not the actual root tip. Interesting. Sorry, bear with me while I find the thing here. Oh my goodness. I forget it's opposite when you're looking in. Well, it looks like my lens is real dirty. So you can clean your lenses with that orange cloth that came with your microscope. Ooh, did I just randomly find it? I need to clean this real quick. So if you can't find it on the high focus, sometimes it helps to go back to the other focus that you had and find it on there because it's much easier to do. And then switch back to the high focus once you're in. All right, so I found it again on that one. I'm gonna switch back to the high. Oh, I just moved it. This is where stage clips come in handy, but those stage clips are just trash. There we go. Oh yeah. All right, so let me show you guys that. So this is the high power. This is 40 times plus 15 in the eyepiece. Ooh. There it is. Boom. Okay, so you can see all the blue things around the edge, around the holes. Those are the cells. Okay, and the holes, the white parts, the clear parts, those are just like the holes in the root where things are moved through. Okay, so the actual cells are the blue parts around the edges of those. Oh, cool. You should send me this if you get a cool snip. Send me pictures. Awesome. All right, so that's the prepared slide. Now what you're going to do is on your lab, it says to make a wet mount. Okay, so I'm going to show you how to make a wet mount. So you can use, the first thing we'll do, we'll look at some sea monkey eggs. Maybe there'll be a smashed one or a partially hashed one, but it's not very interesting. And then I'll open my pond water to show you guys. All right, so you need a blank glass slide, okay? If you guys end up don't having these, you can use the other side of the prepared one. So you can put it right here next to it and use that if you need to. All right, so you're gonna have this and you're gonna have a slide cover. Then you're gonna have something, your little dropper doodad. You're gonna suck up some of your eggs here, maybe some shrimpies. I don't see any shrimpies. All right, I'm gonna switch to this one. Okay, let me switch this way. All right, so here's your slide. Okay, to make the wet mount, you're going to put a drop or two on your slide. Looks like I got a lot of eggs in that, so I'm just going to do one drop. And then you're going to take a glass slide cover. Be careful, these are glass and they will cut you if you're not careful. So this doodaddy, okay, you're going to touch the edge of it to the water edge so that it moves along the edge of it, and then you just drop it, okay? And that is what gets all of the air bubbles to not form, but be careful because it will slip slide around, okay? All right, then you're gonna put that on your microscope, but first you wanna go back to the lowest power. And let's see if we can, ooh, like right when I looked in, it's even focused and everything. So there's, the eggs are like big black dots because they're so thick, the light's not coming through them. So they're not very cool. But the one next to it looks like a partially hatched one. Nope, it's my finger. Almost. There we go. So the blobby one is like a partially hatched one that must not have made it. Looks like its egg casing is next to it. And then the black circles are the eggs for the sea monkeys that aren't hatched yet. Okay, so that's cool, but still not that cool because it's not moving, right? So you can move it around, see if you can find, oh, that looks like a chunk of something. Just a bunch of eggs. Oh, there's a partially, ooh, what's that? Let's put that there and then move to the next power. I'm gonna have to do two hands. All right, so now I'm on the next power up. Come on, come on, come on. I was so sad when Amazon emailed me and said, your camera is showing up on Friday. I was like, oh, man. All right, so that's higher power, so just a little bit bigger. And then that's just a chunk of junk at the bottom. It doesn't really look like anything to me. So that's cool. Those are the eggs. Now I want to do another wet mount with my pond water. So you're going to do the same thing. So first of all, let me show you this pond water here. Pretty cool. I always scrape the bottom to get some good stuff. Let's see if there's anything showing up. Just swimming. Just keep swimming. Just keep swimming. Not focusing very well. I can see stuff swimming in there, but it's not showing up on the camera. So let me dig around in there and try to grab something real quick. Oh, there's some swimmers for sure. Oh, they're fast. Okay, I think I got some good stuff. So you're gonna do the same exact thing. Okay, so you're gonna have your slide. With the pond water, anything else you guys wanna look at, you can do multiple drops to get all of the junk on there. Ooh, what's that? And then you can put multiple cover slides on. Something I like to do with pond water too is, sorry, I see a bunch of guys in there, is to get a paper towel. So hold on, I'll be right back. So if you do this, you have a bunch of water on there, you can touch your little towel, paper towel, or I'm using just a trash towel, and suck up some of the water, right? So all the junk stays, but some of the water's gone. So you can do that. Uh-oh, what happened? There it is. All right, so then you take, glass slides never come out for me. Ah. All right, then you take your glass slide again. You touch it to the edge of the water and then just drop it. You're trying to do that to not get as many air bubbles. So then if you're going to do multiples, just do the same thing but on the other side. Okay, so now I have 2 cover slides on there. So let's see if we can find anything. Now the problem with this is that I got water on the underneath side. That's not gonna help me. So I'm gonna dry off the bottom here before I put it on. All right. All right, let's see what we got. I'm gonna go scanning on the low power. Always start there. And oh, it looks like I got some sea monkey eggs in with the pond water. So I see some tiny floaters, but I'd like to find you guys a big guy. All right, here we go. Sorry. When does this class end? 3:05? We've got plenty of time. There we go. Whoop, come on. Where, oh, where is the opening? There it is. All right, so you can see a little guy swimming around in there. There's actually a couple. Ah, come back. There we go. If you guys just watch real closely, those are single-celled organisms swimming around in that pond water I just got. Are you guys able to see that? Cool. All right, I'm gonna go to a higher power, see if we can see the guys swimming even better. Ooh, there's a big one. Come here, big one. The thing with pond water is that it moves. They move. There we go. Cool. There's 2 big guys there swimming around, sucking up food. And then if you watch, you can see little guys coming in too. Oh, no, 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 no. Where to go? There we go. And that big black circle is a brine shrimp egg that accidentally got in there. Trying to do the focus. There we go. Then you can also lower the light on the side of the stage. Sometimes that helps see things better. Oh, cool. Next time you go to the lake, this is what's going to be swimming in your mouth when you swallow lake water. It's okay. You'll be fine. So let's move it around a little bit. See if we can't find anything else here. I just want to talk about how talented I am right now, moving everything and holding the camera. All right, so that's like a big chunk of leaf, it looks like. And there's a little guy swimming underneath it. Oh, there's a fast one. So this lab is mostly about experimenting with your microscope and trying to find, you know, different things, just having fun with it. Oh, there's a big guy under the leaf. Do you guys see him moving? I'm afraid to move. Oh, he keeps going out of floor. Ah! So that was a euglena. Oh, now I totally lost him. No! Anyways, now I'm just kind of having fun. But it's cool what you can find. So you can also do anything under your microscope. You can put, I have a butterfly wing right here. I can show you guys. Let me grab it. So I'll just put it on the slide. This one you're going to want to have a lot of light for. And you can't breathe. You guys can take a piece of plant from your home plants and take a look at those under there. All right, so this is the edge of a butterfly wing. Oh, I almost went right to it, didn't I? Uh-oh. Did I unfocus it? Oh, I lost it. Hold up. Oh, you know what happened? I blew it off. I blew it off of the slide. There we go. So the reason why we had you hatch the brine shrimp is because they're so cool to watch swim around. And I'm really upset that me as a science teacher didn't get any hatched before class. Anyways. Oh, did I blow it off again? Well, there's a piece of a butterfly wing. And, oop, there it is. So there's the edge of the butterfly wing. It's kind of like hairy. It's pretty cool. And then if you move inside more of the wing, you can see kind of how it's furry. Maybe not on this tower. Anyways, that's pretty cool. So what I want you guys to do is have fun with it. I want you guys to find some cool stuff to look at. If you don't have sea monkeys, that's totally fine. Get some other stuff. Go to the pond. Go to an irrigation ditch. Anywhere that has like algae or mucky water. Tommy, you're struggling to get your microscope to work, it looks like. So question 1 and question 2, let me open them real quick. So question 1, how do you calculate magnification on the light microscope? So you calculate it by the lens down here, and then also what is up here, okay? So this is 15 times, and this is 10 times, okay? Then the next question, explain how to focus a microscope using high power lens, includes any safety issues you need to be aware of. So can somebody else answer that one for us as a group? I talked about it a lot. Okay, Tommy, I'll help you here in 1 second. So I'm going to show Tommy how to use the microscope one time all the way through. You guys, I want somebody to answer in the chat box the question number 2. Explain how to focus using the high power lens. All right, Tommy, you ready? Here's your microscope, okay? Make sure that it has the light piece attached. Sometimes they come separately and you gotta push it in there, okay? Make sure that your light is on. You want your light to be on. And then you want to be on the lowest, the shortest lens, okay? And this adjusts up and down to focus. So then you can put your onion tip root on there. And then you just slowly move the lens closer to the stage while you're looking through it, back and forth until you see it. And you also have to make sure it's in view. So it's blue. So you move it around until you find something blue, right? And then you focus it, just barely turning these little knobbies right here. Okay? If you are struggling, like there's not enough light coming through, you can move this toggle switch on the side here. Make sure that it's on the biggest circle to let the most light in. And then you can change that later as you go. Okay, did that help, Tommy? Looks like Alexander put something in. You use the 2 dials on the side to focus, and you can also use the bottom dial to change the light level. Boom, good job. Yay, good. All right, so the answer to question number 2 on your lab report, looks like nobody put it in there in the chat box. So when you're moving to the large, your high focus, high power lens, okay? If you are too close, you can scratch the lens by rubbing it against the slide, or you can break it, okay? So if you're focusing the lens, so see how close it is to the slide right here, okay? Because it's really long. Okay, so if you start getting real close, this lens will touch your slide. So if you're not careful and you just keep twisting and twisting and twisting, you would push right through and break the slide and possibly the lens. Okay, and those are expensive. So that is a safety issue you need to be aware of. So there's your question number 2. Awesome. Any other questions? We have 4 minutes real quick. Any other questions on how to get the microscope to work, what you need to do, what you need to turn in? Everybody's looking good? All right, so again, at the end of class, I'm gonna email out, you can find all this in 105, yes. In 1.05, you'll find the directions as you read through the lesson, but also the links to the lab report and the student guide are in there as well. So those are like the detailed directions all on one page instead of each page of the lesson. But I'm going to email out this recording and some more information to help you guys get this lab done. It is due tomorrow, but I know a lot of people are waiting on supplies and stuff, so I don't take late points off. Okay, but it has to be done by the end of the block. So it's due tomorrow. Try to get it in tomorrow, but technically you have until the 24th. Thank you, Alexander. That's awesome. Yeah, I'll try to find some more cool stuff on my pond water and send you guys some cool stuff. Okay? Do you have to do the lab on your own? I'd like you to try using your microscope because it's cool, but you can sketch the pictures that I showed you guys if you want. Okay? You do need to answer and turn in this lab report paper. This is the required bit. This is what I'm going to grade. Does that make sense? All right, and then real quick before you go, 2 minutes left. You also have your second Flipgrid is due today. If I can open it. Hold on 1 second. So it should be in OLS, but I will also send out the link again in case it doesn't work. Okay. We'll talk about the Wally after my class, okay? I'm almost done. 2 seconds.
I just want to see the camera.
I know, I just got to switch. All right, so here is your Flipgrid number 2. So you have 3 grades in this class right now. You're going to have your Padlet, where you answered the questions about yourself. You're going to have Flipgrid exit ticket 1. You're going to have a Flipgrid exit ticket 2. And then tomorrow you will have your lab report that will be on your grades. I have not entered grades for the first exit ticket because I was giving people time to get that done. But it's the same thing here. Here's the question. Here's a little video you can watch if you don't know the answer. You can also watch your classmates' videos and then you record your own. And I just want to say, you guys, the videos that I got on the Flipgrid exit ticket 1 was really awesome. I loved them. They're fun to watch. I appreciate you guys working so hard. All right, unless you have questions, you guys are free to go. Let me check back on the chat box here. Okay. Alexander, you're funny. Okay, so if you have questions, stick around, put it in the chat box. If you don't, you are free to go and have a good rest of your day. Thanks, guys. Bye, thank you, have a good one. Bye.